Hi, everybody. Welcome back to microbiology. This talk is going to be about chapter 8, which has to do with genetic engineering, essentially. So let's get into this. Here are your learning outcomes for this chapter and what you should be able to answer as far as once we've gone through the chapter. So most genetic engineering, we're going to be talking about things like using our knowledge about DNA and the technology we have developed to understand DNA, using that to identify people from one another, so like forensic evidence, fixing genetic mutations to treat diseases, using CRISPR technology to repair genetic mutations in a heritable way, so that we can therefore no longer have to pass on whatever genetic abnormality we might have to our children. They can have a normal genetic life. And then recombinant DNA technology also allows us to produce human proteins in non-human organisms. So first we have to understand how DNA behaves as far as whenever we will heat it and cool it. Essentially, when you heat DNA, the two strands split apart, the two strands of the double helix, the A's from the T's, those were held together by hydrogen bonds. And those relatively weak bonds, you add heat into that and it will split those molecules apart. And then when you cool it back down, they'll come and match back up together right in the same spots where they would normally have matched up. So that's how that will work. And a lot of this stuff will run off of that principle, not just for like PCR, but also for hybridization and things like that. So let's get into it. First, we have restriction endonucleases. These are enzymes. So enzymes are proteins that are going to help a chemical reaction occur and overcome the activation energy or the energy required in order to achieve that goal of the reaction. So restriction endonucleases, these are enzymes that can recognize DNA, typically in like wild type bacteria. They recognize foreign DNA and cut it up to help get rid of it so it's not ruining, how they're trying to live their lives. Instead, now we have hijacked that system and used them to allow us to cut DNA in specific predictable places. And that helps us either identify somebody, which we'll get into in a moment, or create sticky ends that we can insert new pieces of DNA into those original pieces. So this is often used in plasmids, which are those small circular extra bits of DNA that aren't essential for bacterial to live, but can help them, survive better. A lot of times these restriction endonucleases act at sites that we call palindromes, same forward as they are backwards, like race car, and they'll clip at that, creating a sticky end. What that means is, so sticky end, let me get the pen, I guess, because I always forget that I need the pen. What it means is you have your double-stranded DNA, and those are hydrogen bonds between the A's and the T's and the G's and the C's, right? So we split this. If you split this apart, you'll have one strand and one strand, right? Well, what restriction enzymes do is they will see, again, it won't be race car, right? It'll be like A, T, C, C, T, A. And be able to recognize that and cut it in a way that will allow it to have a sticky end. And then that will leave us with and then the other side will be this is double strand. And this is single strand. So you get the idea. So they would have stuck together. This enzyme is now breaking those phosphodiester bonds between the nucleotides and leaving us with sticky ends. And we can use those sticky ends, whether it was for ATC or whether it's its complement, of on the other side of the TAG. But whatever it is, it will leave us with sticky ends that we can now match up, whether we are genetically engineering something that will match up and can then insert into a plasmid or something like that, or if it's something that just naturally could be cut and then insert on its own. So sticky ends, the whole sequence that the restriction endonucleases can act on is about 10 base pairs, and the ends themselves are going to be, four or five base pairs that we can hybridize or have matching nucleotides with to insert DNA. So here's a better picture of what I'm trying to say. This is the a plasmid, where like the purple represents the original plasmid here. And we have cut it with our restriction endonuclease. Oh, geez. So this one is C tag going this way, and this one is C tag going this way, and also C and T, and this would be going the other way. It would match up the other side, A and G, from to this. So that's how the sticky ends work, and that's how the palindrome sequences work for restriction endonucleases. So they'll cut and create that overhang that allows us to match up nucleotides and then stick them into places where we cut it with the same enzyme. So here we have DNA from organism 1, and we have cut it with, ECO R1 or something like that, some sort of restriction endonuclease. And then we are able to cut DNA from organism 2, which could just be a plasmid, it could be an actual chromosome, and cut it with the same enzyme. And now our piece can be stuck into this one. And there are ways that we can make sure that this happens whenever we do introduce this DNA back into the organism. But we're not going to go into that now. That's something that you would want to learn in the transformation lab, in lab. So this is going to bring us to the concept of restriction fragments and restriction fragment length polymorphisms. Basically, you're cutting something with restriction endonucleases. If I cut all of my DNA with one endonuclease and you cut all of your DNA with the same endonuclease, we would have different patterns of what would come out a lot of the times because our DNA is just a little bit different. We have differences in our genes and all of that. So the differences in the cutting pattern by those restriction endonucleases gives restriction patterns of different lengths for everybody and every organism. So this allows comparison of the DNA between two organisms at a specific site, because you're not looking at the whole genome typically, you're looking at a piece of it. So we can identify people based on similarities. If all of it's the same, and then it's more likely that that would be the killer, right? So if we're going to put stuff in and stick things into the new DNA and insert genes into that new DNA, and we're going to, end up sticking those together on those sticky ends that we used with those restriction endonucleases, then we're going to have to seal everything back together, because the whole point of the restriction endonuclease was to break those phosphodiester bonds. Well, now you're going to have to remake those phosphodiester bonds whenever you've inserted your new DNA, and we use DNA ligase for that. That just seals back up those phosphodiester bonds. Right, and then we have reverse transcriptase. That's another tool that we often use in genetic engineering. This came originally from like HIV, right? And allowed the RNA from HIV and turn that into DNA, right? I think we talked about this already in the virus chapter. So, but yeah, basically turning the RNA into the DNA. That can allow us to turn RNA samples that we got from something and then turn it back into DNA. For PCR or for cloning purposes. This allows us also to work with something like complementary DNA. Remember how eukaryotes have introns that need to get spliced out from the mRNA in order for it to make the usable protein products by the ribosome. So what we have, after we have spliced out our introns, and now we have our exons sealed up all together, our usable exons. This is our working mRNA. It's already been edited. So we can extract that messenger RNA from cells and isolate it, and then use reverse transcriptase to make the DNA version that does not have any of those introns in it. And this is a double-stranded cDNA. cDNA is basically just cloned DNA, cloned from that messenger RNA. So that way bacteria can read it, because bacteria won't be able to read human genes, right? Because we have introns in them. We need to get rid of those. And bacteria can't splice those out. They're not capable of it. All right, CRISPR. Well, I know a lot of you have heard of CRISPR. Everybody keeps asking me about CRISPR, or like what I think about CRISPR or whatever. I mean, it's a tool that is useful in genetic engineering. It stands for clustered, regularly interspaced, short palindromic repeats. These are short bits of DNA with repeating nucleotides in bacteria and archaea. And then normally the enzyme in that group of genes will cut out, so Cas9 in the CRISPR region will cut out foreign DNA, typically left behind by invading bacteriophages or plasmids that it recognizes. And so now we can exploit that system to cut an organism pretty much very specifically exactly where we want to. And that would allow us to remove genes, as well as hopefully eventually also insert genes so we can remove a mutated gene and replace it with a working one. That's the idea, right? So that's the CRISPR system. So polymerase chain reaction, our best tool that we have ever invented as far as genetic engineering is going. This is the polymerase chain reaction that allows us to replicate target DNA from a few copies to billions in just a few hours. It can detect it can detect cancer from one single cell sample, and then can also diagnose infection from a single gene copy from the organism that could be causing the infection. So PCR will use the same events that we would see in DNA replication. We're going to open the double helix. We're going to expose each strand and use each strand as a template. We'll add it in primers because DNA polymerase has to build off of a primer. And then we will have the actual action of the DNA polymerase. Typically, you need to, number one, I want to be clear about this. You have to know the sequence, not have an idea of where or what it could be. You have to actually know the exact sequence that you're trying to work with. So your primers have to match exactly that specific thing. If they don't match, you're not going to be having PCR happen. So you have to have primers available that actually sequence out specific known gene for, salmonella or something like that, if you're trying to test for that. So you have to actually have primers for those things. They have to be known. They have to be known. Okay. You can't just say, oh, we'll just PCR it and find out what it is. That's not how PCR works. It doesn't answer questions like that. You have to have the question that is more specific, that you would test for, oh, I think this is E. coli. And then we'll run the program for E. coli and add the primers for E. coli, so that's the idea. That's what I'm trying to get at here. So you have to know what primers to use and what sequence those primers should be in order to actually multiply or replicate that gene in the PCR machine. It's not magic. So DNA polymerases allow us to replicate DNA. So we're just going to be adding nucleotides on in the 5' to 3' direction. Because we have high temperatures going on in PCR, we need high temperatures in order to denature the DNA itself and split those strands apart, which we talked about previously in this chapter. Then we need a DNA polymerase that adds on the nucleotides. We need one that can withstand that high heat. It can't be denatured. A lot of times proteins can be denatured by heat as well. So you want to be sure this now we have a protein, an enzyme, DNA polymerase that can withstand that heat. And so we have TAC polymerase, which is Thermus aquaticus. So that is something that grows in the deep sea vents. And we also have another vent polymerase, and that is from Thermococcus littoralis. So both of those are useful as far as PCR goes, as DNA polymerases that can withstand heat. So this is the steps involved, the 3 basic steps that are involved in PCR. First, we have denaturation, which is the splitting apart of our DNA double strand into 2 separate strands. So that's step number 1. Step number 2, we're going to prime. You have to cool it down a little bit to get those primers to stick. So super hot here. Cool it to allow the primers to stick, and then at the extension temperature, we would have a little bit warmer temperature, usually about 72 degrees Celsius, where that thermus aquaticus or whatever DNA polymerase can work at the higher temperatures. But anyways, then we have extension, and that is just starting from those primers, adding on nucleotides in the 5' to 3' direction. And then just, okay, so then we added our nucleotides and we made our copies, and we made this copy, and then we can go through the whole thing all over again. And we'll do that over and over and over and over. So one double-stranded sample becomes 2 double-stranded samples, 2 double-stranded samples becomes 4 double-stranded samples. 4 becomes 8, and so on and so forth with each cycle. So each cycle will amplify the DNA and allow us to create, copies. So real-time PCR is a procedure that allows us to detect products while they're being made, instead of waiting until the end to see when it's done. A lot of times these will release some sort of like fluorescent color as we're using specific nucleotides. And so you can actually measure the progress of that along while that's happening. So that's real-time PCR, or RT-PCR. PCR can also be adapted to analyze RNA by converting it back to DNA using reverse transcriptase, right? And making the cloned DNA. Now, recombinant DNA technology is literally referring to removing genetic material from one organism and combining it with another organism. And that would be therefore generating genetic clones. Something is carrying a gene that it wouldn't normally have had in, its cell. So cloning, here we have removal of a selected gene from an animal, plant, or microorganism. That's the donor. The gene is inserted into a vector. That's basically the bit of DNA that is going to house that gene, for example. So usually it's going to be a plasmid. Sometimes it's a virus. And then that will be inserted into the host. So oftentimes that's going to be bacteria. It could be yeast. It just depends on what you're trying to make and what makes sense as far as reading that gene and producing whatever product you're trying to make, whatever makes the most sense for that. The gene is then translated. So we'll go from DNA to RNA to protein. And then the protein product will be produced by that organism that has that, new gene. So we will, DNA can be removed from cells and separated into fragments as well by the endonucleases, and then synthesized messenger RNA transcripts can be gene can be synthesized. Okay, so right, we've already talked about that. So I don't know why this keeps popping up. Okay, so basically the common strategies, I think we already mentioned these, but cutting things up using enzymes, making copies of a messenger RNA into cloned DNA and putting that into other vectors. And then we also have genes being amplified by PCR so that we can increase the amount of DNA that we have to work with. So genomic libraries, this is basically taking genes out of a, host organism, an original organism, taking genes from that, popping bits of those genes into vectors of some kind, and then we can grow those up, those vector cells that we place those genes into, grow those up in a culture, and those could be, representative of those genes, and we can have genes from every single gene from each organism. So that's a genomic library. Okay, a cloning vector. These are, we've already talked about plasmids. These are probably the most commonly used. These plasmids are going to be small little circular bits of DNA. They're very well characterized, meaning that we know pretty much every gene that's going on in there, and we know essentially what would be expected as products from this plasmid. Anyways, and then we can insert genes into these and then put those plasmids back into a host cell, a competent host cell, through transformation, which we've already learned about in the genetics chapter. So they can also confer antibiotic resistance and things like that. And that can be important because if you have a target cell that is just plain old E. coli, not antibiotic resistant, and now you want to put a plasmid into that, put a gene into a plasmid that you're going to put into the E. coli, Well, how are you going to know that it got in there? So you can have antibiotic resistance in there as well, so that you can select for it. So the cells that take up the plasmids that have antibiotic resistance in them will also have your gene. So you can grow them now on antibiotic plates. And then after growing them on those antibiotic plates, the only ones that would have been able to grow would have been the ones that have the plasmid and therefore the gene that you're trying to put in. So that's one way you can select for that. So anyways, that's the usefulness as far as that antibiotic resistance goes with plasmids and cloning. We can also introduce DNA with bacteriophages. That's another way we can introduce new DNA into bacteria. I mean, remember, bacteriophages are the viruses that infect bacteria, and they'll inject their genomes straight into the bacterial cells. So every plasmid, this is a plasmid, this is PUC19, P is for plasmid, don't worry about the UC19 part, but this is a plasmid, and this is a cloning vector. And essentially it has a site in it that we call the multiple cloning site. And it has all these different places where you can cut with a restriction enzyme. So whatever your gene of interest, whatever you designed that cloned gene to have as sticky ends as though it were cut with EcoR1, for example, the top one, you can cut this with the same one and then be able to stick that into the spot in the plasmid that you were hoping to stick it into. The actual origin of replication, they all have to have an origin replication of the actual plasmid itself, so that whenever you grow these up in culture, they can be multiplied, you know. The cells that you're using, you would have to be sure that they can accept DNA that is this and that size. Some bacteria are very picky as well as far as the size goes that they're able to take up. And like we were saying, a lot of them often confer drug resistance for selection purposes. You typically, for your cloning host, you're going to want a host that has a rapid turnover or fast growth rate, so you can have your product made fast, can grow in large quantities using culture methods, non-pathogenic, so you don't want making people sick all over the place, all over the time. The genome that is well mapped and well understood, maintains the genes through multiple generations, can accept that plasmid or that bacteriophage DNA through as a vector, and then they can express high yields of proteins, so of whatever your gene of interest is, because you want a lot of it made, right? That's the whole point you're doing it. So there is interferon. This is alpha 2A interferon, also known as roferon A, that they use to treat hairy cell leukemia. This is transformed, but basically cancerous. They're lymphocytes technically, but they're B cells. The B cells are the ones that produce your antibody. And Kaposi's sarcoma, which is associated with HIV infection. They're able to use recombinant version of this interferon to stimulate the cells of these people's bodies so that they will attack the cancer cells, essentially. Okay, so here is the idea of what you would need for cloning. You need the gene you're interested in, and you need the vector you're going to put it in. So put your gene into a plasmid, for example. The plasmid is going to have a lot of useful aspects to it that you need to actually you can't just have a floating little piece of DNA. You need it to have other things going on with it that are going to allow you to, multiply and everything like that. So, but yeah, this is the idea. You're going to cut with your enzyme, and you're going to do that with your plasmid as well as your gene of interest, and then you'll have your sticky ends. So here is a sticky end sticking onto this thing, and here is a sticky end sticking onto that thing, and that's how we'll be inserting our little piece of DNA, because that's how this went. Original plasmid, and then we cut it with restriction enzymes, made a sticky ends, and then we stuck in our little special bit of our DNA that we've treated with the same enzyme. And now we can ligate those together, seal it back together with ligase, and then put it into a host cell of some kind, right? That's the idea of how that works. And then after we have put it into the cell, that cell will go ahead and read that donor gene and make messenger RNA. So we'll transcribe the DNA. It will make the messenger RNA, translate that into a protein product, and then that can be collected from that flask as it is being produced by the bacteria, and then we can purify that protein out and then collect it for whatever studying or production purposes. Synthetic biology is talking about the creation of new biological molecules and organisms completely from scratch. You can do this using just the nucleotides by themselves. So this has been done, and we have created living cells to do using this concept, using just chemicals. When we are looking at like mapping and profiling DNA, these are essential as far as comparing people, whether we're talking about the exact sequence, genetic sequence of something. We can also be talking about what the pattern looks like when we cut with a specific enzyme, for example. So we can use that to identify or tell people apart. But one of the things you have to do in order to do that is run it out on a gel. And that gives you those bands that you are typically used to seeing with a DNA analysis. So basically you have agarose gel. It's a gel, I don't know, a semi-solid jelly type stuff. And the larger pieces, the larger DNA fragments, they get caught in the gel. So they get slowed down, and the smaller ones can travel faster. So as you are putting electricity through the gel in the fluid that it is sitting in, that electricity is going to push DNA, which has a charge, negative charge on it, through from its starting origin in the wells that you put it in, and then towards the end. And it's at the bottom of that gel is where the smaller pieces of the DNA are going to be, and the larger one's going to be closer to the top. And that is based on like the size of the DNA, and the position on the gel is relative to that. And we can even use ladder that we know has known size of fragments to know like which size each band would be. So this is an example of a gel that would have run out on gel electrophoresis that had been, we're looking at all of these little pieces that look, each band is a different size of, all of these are different sizes of DNA fragments. So, and this is our known ladder over here on the side. Let me erase this so you can see it. So we know what these are. And then these are the shorter bits over here. And these are the longer ones, larger pieces. And that's how that works. Okay, nucleic acid hybridization and probes. This is essentially using those matching up of the A to the T and the G to the C, matching those back up with little pieces of known pieces of DNA, bits of DNA, to see if this keeps sticking in my face, to see if the gene of interest or whatever is in that particular location. Like you're on your plate, like your bacterial plate or whatever it is. So essentially it's going to allow us to match up gene sequences with one another. Okay, it's called hybridization because we're going to bring the pieces of the DNA, single strand DNA, back together from a probe and your original source that you're checking out to see if it's there. So that's the idea of how that would work. Yeah, and they have enzyme linked probes that can lead to pigment change color. Essentially, if the enzyme is there, that means the probes hybridized and it matched. It was a match, and it can lead to a color change whenever the enzyme acts on whatever thing is the substrate. So, all right, so using gene probes, this can help diagnosis of an infection, and it can also allow for identifying culture of unknown bacteria or virus, and that's what the usefulness would be for that. So the actual nucleic acid hybridization test itself doesn't require any electrophoresis. We have our probes, our little mini bits of DNA, and then those single-stranded probes will block with single-stranded probes will combine with their match on whatever gene you're looking for. And if it does match, that means that, you can identify those colonies, bacterial colonies, for example, as having the gene of interest. And these are the ones that, took up your plasmid or contain this and that gene for a pathogen or whatever it is that you're trying to look at. Okay, fluorescent in situ hybridization, also known as FISH. These are probes that can be used on intact cells without having to break the cells open at all to get to that DNA. So we can actually use intact cells. So we can identify bacteria living in their natural habitats without having to culture them. So that's a useful thing. It's also a good way to identify genes on chromosomes. So yeah, we can detect specific RNAs within tissue to see if the particular product is being, made, transcribed. Right, so it's important to understand the sizes of DNA, and basically what I want you guys to know is that we can talk about it in megabases, kilobases, and just base pairs, right? So a BP or base pair is one single nucleotide pair. When we look at palindromic sequences, those are going to be 4 to 10 base pair long sequences. When we look at E. coli's entire, when we look at E. coli, the average gene of E. coli, they're typically 1,000 base pairs. This also would be a kilobase, 1 kilobase, that you guys know what that means. And a megabase would be like 1 million bases, okay? And then she's just comparing the size of things from here on out. But you guys do need to know the difference between base pair, kilobases, and megabases, so that if I'm talking about base pairs, if I were to ask you, which one is bigger, and your choices were which one's the biggest. Okay, if I were to ask you which one is the biggest, you would pick the bottom one because that's 5 megabases, so 5 million bases. This one here is only 5,000, but it would be the next big. So this would be number 2. The next big after that is this one, number 3, and the smallest is this one, number 4, right? So that's the type of thing that I want you guys to be able to recognize whenever we are taking our exams. Okay. DNA fingerprinting. This is just using forensic science that relies on this genetic engineering type of science to use cuts in DNA and then compare the results based on the differences between different people using various enzymes to see if if this blood sample is your blood or somebody else's blood. And it's a lot easier to use that than sequencing an entire genome or something like that. So, right. DNA will be cut at specific sites based on the sequence of those sites. So based on the actual sequence of every single gene, potentially in someone, that'll be a unique cut and yielding a unique product for every single person using even just one endonuclease, right? Then we can run those out on the gel and compare what the little lines on the gel look like. And if the blood sample looks the same as your sample, then you're the killer. Or if the blood sample doesn't look the same as my sample, that is not likely that I would be the killer. So this is just showing us a gel and the different subjects that they would be looking at. So they have the marker here, we have our victim here, and then we have collected evidence. This is evidence that we collected at the actual crime scene, perhaps. And you can see here that the suspect that you are concerned about, suspect 1, matches up with the same as the evidences that we are seeing in that location, where suspect 2, the other person we were concerned about, does not match. So that is just going to be comparing those two and allowing us to look at those to see if they match up in a predictable match up in those using the gel electrophoresis based on the sizes of the fragments that are made using those enzymes. Okay. Sequence maps. These are detailed maps of genomes. They give exact order of bases in plasmid, chromosome, or the genome. Right. So humans apparently share 80% of their genetic codes with mice, 60% with rice, and 30% with worm C. elegans. Right. So I don't know. So it's just basically comparing at the nucleotide level similarities and what gene is where and what the genetic sequence is for that gene, and comparison between other organisms that have that same gene or essentially that same gene, and their differences and similarities. So that's the idea with that. So high throughput genome sequencing, this is doing like essentially shotgun sequencing to try to figure out the overall sequence of organism in a faster amount of time than what we typically would do if we were doing this by hand and individually. So whenever we are talking about managing high throughput data, when we're getting all of this information, we have to have a way to analyze and classify the genes that we are identifying through this sequencing process, determine the protein sequences that would be resulting from these genes and the function of those genes, the proteins that are made from the genes, right? What do those proteins do? Right, so in determining the functional information of that gene sequence and trying to determine what protein it's going to make and what its usefulness is, that can be tricky, but all we really have to go off of is identification of other genes in other organisms, and then basing our results for people on, or not just people, but whatever we're studying based on what is known, right? So well annotated genomes can provide us abilities to see what our normal cells would be doing, what ones look like they would be causing disease, what mutations are involved in causing blah 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 disease or susceptibility to disease, development overall as we're getting older, and then aging as even older, and start declining in our health and everything like that. So this can give us a greater understanding of all of these things, dealing with just the human body itself. So single nucleotide polymorphism this is just referring to change in one single nucleotide that has caused a devastating or detrimental effect to people. So in general, the difference from person to person in some of these, I guess, don't always have to cause bad effects. I shouldn't have said it that way. Some of them cause detrimental effects. Some of them are just changes from what is normal or what is considered normal. So anytime that we have that, that's a single nucleotide polymorphism. It's just one nucleotide that's been changed. And we have tens of thousands of these differences at a single locus that can exist throughout the genome. The human genome has about 10 million single nucleotide polymorphisms, so single change in that nucleotide. And then, they're trying to map those differences to see which ones put people at greater risk for specific disease. If they understand that single change for that specific location, what is it going to make someone more likely to have blah, blah, blah disease or not? So that's the idea of how that would work. So yes, the ones that are detrimental are the ones that we are the most interested in, but you can have some that don't have necessarily any effect. So examples of some of these single nucleotide polymorphisms associated with diseases, we have the normal DNA, quote unquote, is GCT. And then now we have a CGA that matches up with that, right? And this is what it would code for. This would be your arginine here. So it would be arginine, glycine. Well, this person down here below has GTT instead of GCT. And then its matchup is a CAA as a result. And that would end up giving us glutamine, GLN, whatever that stands for, that particular amino acid. Sorry, I don't have them all memorized. But yeah, so GLN, this is going to, glutamine. Okay, well, it says it right in front of me. So now it codes for glutamine instead of arginine, which is what we saw up here. And that's going to change the shape of your protein, and your protein might not be able to function the way that it was supposed to. So that is an example of the actual literal change that would be going on there. So as far as what these people experience, this has to deal with, or it was in the previous slide, thrombophilia. Okay, so increased clotting, so developing increased clots, and that could, of course, put you at risk for stroke or something like that. But yeah, excessive clotting just due to that one amino acid change from that one nucleotide change. That's pretty impressive, and the importance that one nucleotide can have. So we do also have DNA microarray analysis. This allows biologists to view the expression of genes in any cell all at once, all out on these large plates, and allows for hybridization of like RNA to other RNA that's already on these plates to see, you know, which RNAs are being made. That's essentially the idea of microarray analysis. That allows us to determine what sort of genes can be are being expressed or being made actively in the cell. This can let doctors and apparently pharmacologists look at RNAs that are being made like in cancer patients and help target genes specifically that would be involved in having, you know, cancer. With regards to recombinant DNA technology and other uses, we can develop, you know, we can have the development of recombinant organisms, so the ones that are carrying genes that we're interested in, sources of protein products, having organisms work as food for us or create food for us. We can study the effects of certain nucleotide sequences and changing them and investigate things in other organisms so that, you know, we don't have to do it in our own bodies. We can manufacture medications that we wouldn't be able to have manufactured otherwise, so we can have organisms do that for us. And then manufacture of hormones and enzymes that we can use for people and their health. This includes things like interferons, interleukins, tumor necrosis factor. This includes like Remicade and Humira. All of these are things that we can make by recombinant DNA technology. So these are all related to the immune system and can be used to fight infections as well as autoimmune diseases and cancers. And we can make erythropoietin that stimulates bone marrow to treat certain forms of anemia to make, you know, more red blood cells, and then growth hormone for, you know, literally helping people grow when they don't have that hormone available naturally in their bodies, telling them to grow. All right, we can also use this for development of vaccines, which, you know, we talk about in some of the later chapters. And then factor 8 here, this is a replacement for blood clotting for those people who have hemophilia, so they bleed too much, you know, bleed too easily and bruise too easily. So transgenic organisms are the same exact thing as genetically modified organisms, same thing. Okay, these are recombinant organisms where we have a gene from one organism put into another organism that doesn't normally have that gene. Okay, so foreign genes have been inserted through recombinant DNA using creating GMOs into microbes, plants, and animals. We've successfully done 3. So yeah, so I'm not going to go into the details of some examples, but these are some examples of microbes that have successfully been used, inserted some special plasmids into them, and therefore had them create specific products that we can, you know, use for agriculture or something like that. So so as far as genetically engineered plants goes, we have plants that are resistant to herbicides and pesticides, and they'll be resistant to certain viruses. And now they call them they can, you know, lead to super weeds that can't be killed by anything. You might think that that's great for a crop, but what happens if we have the genes being swapped to certain, you know, non-genetically engineered plants, and now we can pass on essentially the resistance to these things and causing problems in the ecosystem as a result. There's a problem with that. Gene therapy refers to repairing faulty genes in humans that could be suffering a fatal or debilitating disease. Essentially, we can provide a cure which is permanent to that person who's receiving the treatment. Right, and we can make it so that it can be heritable in some cases. So what we can do, one strategy is to take a retrovirus or an adenovirus, remove tissue from the patient, and like this is talking about like bone marrow, okay? Incubating the bone marrow with those viruses, so the viruses infect that bone marrow, and then putting that back into the patient And then now those viruses that were introduced can pass it on to all of the genes in that person's body, hopefully. And that's the idea of how it should work, or how it should be transmitted, and then therefore causing the genetic change in the patient, because we have taken the bad things out of those viruses and put in the gene that that person was wrong, it was bad, it was mutated for that person. So for ex vivo gene therapy, that is exactly what I was just explaining to you. Putting a healthy gene into a virus, putting that virus into a bone marrow, and then putting that bone marrow back into the person and hoping that it gets incorporated into their genome, and it can be permanent. There are gene therapy treatments for 13 different diseases that are available. Some of them include spinal muscular atrophy, inherited vision loss, acute lymphoblastic gene, I guess that's leukemia or something like that. Anyways, I didn't type that, so whatever. So somatic gene therapy is going to be changing their actual like body cells, but it's not heritable. It's not going to have anything to do with the germline or the gametes, the sperm or the egg, right? Whenever we do treatment that affects the germline, then we are introducing those genes into the egg, the sperm, the embryo, whatever it is, and that can lead to it being heritable to the next generation after that. We can also use microRNAs to silence genes. Those are just RNAs that match up to certain parts of genes so that they can't be like transcribed properly anymore, to block the production of whatever that bad thing is that you're trying to make. So we can use that to control expression as well. We've used this for, I guess, looking at stopping growth of cancers. When in studies done in mice, in monkeys, we have lowered cholesterol levels, and then chimps, we have multiplication. Okay, it was just weird. It was not worded as the same as the other ones. Hep C was essentially blocked in chimpanzees using microRNAs. I don't know why it was worded differently than the other ones. Okay. Yeah, so interfering RNAs like the microRNAs, these guys, for humans in general, what we have done, they've been successful as far as nasal sprays to silence genes from RSV, so respiratory syncytial virus, which can be pretty bad for little babies and stuff. So that can eliminate infection and symptoms associated with RSV. So 44% of those using that were infected, compared to 71% of the control substance. So we're able to prevent, growth, spread of this infection using those silencing RNAs. Going back to CRISPR and Cas9, this is naturally occurring, right? We already mentioned that. Bacteria using this as a means for defense against foreign DNA. We can use that to precisely edit genes in any organism, which is very useful as far as creating changes potentially at the somatic level, but hopefully at the germline level to be heritable later on in certain faulty genes. So, all right, so this is the end of this chapter. What are some concerns with regards to altering the genetic code of a patient, even when changing a single known genetic mutation? So think about that and what the concerns would be. And then I'll see you guys for the next chapter. And goodbye for now.